how malaria is a medical emergency. Okay, so right, one of the interesting things that has recently come up during the COVID pandemic is a realization that COVID may not actually be the biggest monster that is spreading. There's actually TB, HIV, and malaria, which has been left somewhat neglected as a result. And the social distancing measures have put in place barriers for people which would otherwise need to travel in order to obtain diagnosis or drugs in order to treat these infectious diseases. And so recognizing that a longer person takes to be diagnosed, the later treatment begins, the more likely he is to get really sick from the disease and possibly die from it. Moreover, you also see increase in transmission because of this lag. So it's a disease that we cannot afford to have any delays for. And in this next couple of slides, I'll try and explain why. So Prof. Tan has really brought up to the life cycle and how it was discovered. But I just want to point your attention to the area in red, the erythrocytic cycle. This is the only stage which causes disease, where the fevers manifest, where you develop complications. It's on this particular part of the life cycle. And if you look at the clinical symptoms, they can be very easily missed. They can be construed to be COVID-like symptoms or general infection. So the symptoms are not specific at all. And unless doctors know what to look out for, it can be misdiagnosed quite easily, right? And the realization that malaria is able to kill very quickly is one which really should catch our attention. Right? Traditionally, diagnosis is done by microscopy, and it's heavily reliant on rather tedious and slow methods in order for diagnosis to be done. So I have a picture here from the Malaria Consortium showing that how the disease can progress very quickly. So you get fever and sweat and chills, headaches, very quickly progressing towards convulsions, coma, and death within 24 to 36 hours after a person can be coming in to the clinic. So why is there a reason for this rush? Well, there are a number of reasons for, at least for falciparum malaria. The life cycle is about 48 hours, and a single parasite or a single infected erythrocyte is able to produce 20 to 30 or so parasites each time. And so these parasites, moreover, are able to infect erythrocytes of various stages. So pretty much any red blood cell in your body is vulnerable to attack. This leads to a very high parasite density, parasitemia, and this high biomass of parasites in our bodies causes hypoglycemia. The parasite is very hungry for sugar, so it takes in all that glucose and readily converts it to lactic acid. And so the increase in acid in our blood also yields significant pathologies, right? Of course, with such a heavy parasite burden, you expect massive inflammation, leading to symptoms like respiratory distress syndrome, acute kidney injury, and others. And I'd like to point out your attention to the last point, how infected falciparum cells tend to sequester. I explained that in a little bit in the next slide, but essentially this gives rise to the most severe forms of malaria, including cerebral malaria, placental malaria, lactic acidosis, and such. So what is sequestration? In the talk before, we learned how Leveran found gametocytes, right? So the reason why he found gametocytes, even though they're only a tiny fraction of the parasites in our body, is because the other stages, namely the trophozoites and trizones, are absent from peripheral blood most of the time. And the reason for their sequestration, or the reason why they're trying to avoid being in circulation, is because they're trying to stay away from the spleen. In the spleen, red blood cells that are unfit, that have parasites growing in them, will be filtered out, will be destroyed. So in order to evade the spleen, these parasites employ the strategy of hiding out inside our blood vessels. So there are two mechanisms for this. One is called rosetting, and one is called cytoadhesion. So in this diagram over here, you'll see somewhere in the middle, you see a ball of cells surrounding red blood cells surrounding an infected red blood cell. Okay, so this is called a rosette. And cytoadhesion is when an infected red blood cell just directly binds onto the endothelium, leading to an eventual occlusion of these very fine capillaries. Okay, so it sticks. So let me try and get rid of this high high protein control, right? Perfect. Okay, so it sticks. So what? So I've got a photo here of a brain section from someone who's died of cerebral malaria, right? This is a young child who didn't make it. And when they sectioned his brain, you can see very clearly capillaries in his brain being clogged up with infected red blood cells. So you can see, I think, very clearly in this photo, you can make out the shape of the red blood cells. And the little dot that you see here is the malaria pigment, hemozoin. And so that's how you tell that these are the infected red blood cells. And you can see they're really just lining up on these capillaries, and you see this everywhere in your body where you've got very fine blood vessels. So in your retina, for example, you're able to see a huge accumulation of these purple spotted infected red blood cells. And right in the middle, you can see there's a little bit of uninfected red blood cells that probably still can stream by, but the cross section of the capillary is massively reduced. Right, and incidentally, looking at the retina of infected patients is one way that you can tell if a patient is infected and is in a very bad state. All right, so in terms of rosetting, it looks a little different. So I've got here some acridine orange as a fluorescent stain that stains the parasite green. And all around, you see these uninfected red blood cells forming, at least on the picture on the right, something that looks like a flower petals surrounding the center, right? But if the binding is very compact, it can form a tight ball inside which the infected erythrocyte hides, right? So one mechanism, one benefit to the parasite of rosetting is that it reduces antibody binding to some of these antigens. So that's one of the kind of biological basis for this. Now, if we turn our attention a little bit away from the pathobiology of the parasite, it's interesting to explore how Singapore's history has evolved in response to this disease. So malaria first became documented in Singapore around 1880, and in the early 1900s, we were seeing about 2,000 deaths annually from malaria within Singapore. Right, the peak was in 1911 when we had almost 3,000 people dead, and a result of that was an anti-malaria committee set up by Sir Malcolm Watson. And he introduced an anti-malarial drainage system and oiling program. All right, so if you want to find out why Singapore doesn't have malaria today, these are some of the main reasons, right? You've got an anti-malarial drainage system which removes all the stagnant water from these waterlogged areas, so the vector cannot breed. Oiling program like what we have today still, you have people going around to the drains and pouring kerosene over it, and so that kills the larva stages of mosquitoes. Right, but during the Second World War, when Singapore was occupied, these drains were not properly maintained. And so there was a resurgence in malaria cases, leading to, again, about 3,000 deaths in 1945 alone. So post-war, control measures were intensified, and we entered a season where there was practically no local transmission of malaria between 1955 to 1963. But there were occasional outbreaks after that, leading to stepping up of measures that were taken. So malaria was made a notifiable disease. We recognized that chloroquine-resistant parasites had arrived on our shore. And we also recognized that a lot of the times we get malaria coming to Singapore because people who are infected in their home countries come here as migrant laborers at that time. And because the vector is still around and our population is not immune at all, there's a high rate of local transmission. So as a result, a new control strategy was implemented in 1975 to efficiently identify the foci of transmission, the people who are infected, ring fencing everybody around them and getting them tested, and aggressive epidemic control. So weekly oiling, spraying of insecticides in the workers' dorms, to the point where the vector has been virtually eradicated, with only 13% of our land being able to support vector breeding. Right. So with these sustained improvements, Singapore was eventually declared malaria-free in the year I was born, 1982. So since then, the malaria that we get is entirely imported. We see about 200 to 300 cases a year. And interesting point to note, between 1975 and 1982, Vivax was clearly the dominant species circulating, and there were 10 deaths, all attributed to falciparum malaria, which I had just now cued you in about. Right, so effective disease surveillance really comes down to experience and skilled diagnostic lab technicians. So we have sitting right in the front as one of our VIPs, Mr. Rama, who used to work in the Malaria Reference Center. And so as an impressionable undergraduate student at the time, I came to know about Mr. Rama and saw how he very diligently worked hours at end in front of a microscope to diagnose people and find out which species they're being affected with. Right. So in 2015, the Straits Times also wrote about another guy called Kihar Singh, who is a public health official. And so he was also involved in controlling the malaria outbreak in 1974, as well as subsequently controlling SARS and other infectious diseases. So because of these unsung heroes, we are where we are today and enjoy a malaria-free environment. So that begs the question, why should we, living here, care about this disease, especially if it's completely preventable and treatable? But bear in mind, 150 people died since we started our course this morning. They died of malaria, right? A week ago, there were 8,000. Since a week ago, there had been about 8,250 people who passed away, most of them being children, right? So these are very alarming facts, and should we really be in a place to ask whether we should care? And when we do care, are we going to be too late, right? So Singapore is situated near a hotspot from where malaria drug resistance emerges, and the vector, Anopheles mosquitoes, still reside within our habitat, and especially as Singaporeans are promoting a more green environment and are also making forays into these forested areas more and more. I think interactions with Anopheles vectors is almost sure to go up, right? We still have no immunity to this disease, which is very foreign to our systems. There've also been evidence of diabetes and obesity being risk factors for someone coming down with malaria getting severe forms of the disease. So something for us in Singapore to bear in mind. And with the advent of artemisinin resistant strains cropping up in Southeast Asian regions, recognizing that there are little alternatives is very sobering. So our Singapore story today, there are still occasional recent outbreaks. 2003, 2009, for example, we've got a mixture of Falciparum, Vivax, and Nolazai. Nolazai being the zoonotic form that we'll hear about shortly. So again, the vector is still present, but because we have a very active surveillance system where MOH is notified if there's any malaria, and we'll take measures to make sure that that infected person is not only treated, but his area is safe, that there are no Anopheles mosquitoes to transmit the disease, right? More Nolazai later, and relatively recently, our army boys have also stopped taking anti-malarial pills during their time at Pulau Tekong, and so if we ever have to go into a jungle to fight a war, we might need to have some stash of this ready somewhere. Right, so Straits Times had reported a couple of years ago how malaria-carrying parasites are now resistant to artemisinins, and so there was a bit of a scare in Singapore when you hear things about super malaria happening at your doorstep. And in a 2017 report, it says that Singapore continues to remain vulnerable, but rapid spread within our shores is unlikely due to the limited presence of a vector. And MOH quickly stepped up to say that no super malaria strains reported in Singapore. A couple of years later, 2019, there was a big conference in Singapore, and there was a co-chair of the Lancet Commission who said that we as a country have an important role to play in investing in regional initiatives to target malaria, and also in helping our neighboring countries to eliminate the disease. So we had our Foreign Minister, Vivian Balakrishnan, who emphasized that Singapore is going to do our part, right? We're going to play an active role in tackling complex global challenges like malaria, and of course, now with COVID-19 as well. And he made a statement pledging support for worthy initiatives like this to make the world a healthier place for everyone. So politically, and I think individually, there is a reason for us to play our part and make things a little bit better. So the reason for why we should be involved in malaria is as simply summed up as this. I am my brother's keeper. We are responsible for each other's well-being as fellow human beings. And a wise saying once put it this way: cast your bread upon the waters, or be generous. All right, be generous towards others, and after many days, you'll find it again. Right? You can call it karma, you can call it what you want, but it's a simple principle, and I think this is the reason why we really need to be heavily involved and invested in malaria research, education, and even more on the ground practical um, uh, initiatives to tackle this disease. All right, with that, I end my speech, and I'll hand over the time to Benoit, who will be speaking next about Molazide. Thank you.